Uh, yes, Movani, you can start. Uh, Hello, Dr. Ranudit Chakraborty. Are you
Yes, I'm
listening?
here. Right.
Okay, thank you, sir. So we are starting. Uh,
Yeah.
Movani, start. Hello,
Mormon is not here.
she was here little bit ago. Uh, uh, Shilajit, can you call Movani? Probably she got disconnected. Please call her. Now she is here. So Movani, you start. Okay. Uh, you are not audible, Movani. Movani, you are not audible.
Excuse me, Dr. Das.
Yes.
Your party, all the participants has given their attendance. Means
Yes.
are they?
They are in YouTube live.
Okay. Okay.
Okay. Only the organizers and the speaker. We are here
Okay.
in
I
the.
understand. Right. So,
No, you are not audible. We are not listening to
Monia,
you.
just sign out and sign in once.
Sorry, participants, for some small technological issues. No, Shilajit, it's a problem. Movani is not getting listened.
Monia, please you could say hello. Yeah, you're
No, no.
sounding very low. Hello.
Your sound
Hello.
is yes.
Yeah. It's okay.
Now it's okay.
Now it's okay. Now it's okay.
Good afternoon, everyone. Welcome to the International Webinar Lecture Series organized by the Department of Microbiology, Vijayanagar College, Kolkata, in collaboration with the Internal Quality Assurance Cell. The topic for today's webinar is In Search of Origin, Prevention, and Possible Cure for COVID-19. We have with us three extremely eminent speakers, Dr. Ronodhi Chakraborty, Saint Petri Braid, and Dr. Sayantan Ghosh to enlighten us with their views over this topic in three separate sessions and over 2 days. I thank all my fellow participants today for joining us in this webinar, and I thank our department and faculty for organizing this webinar. Now, I would like to request our respected professor, Dr. Prashanjit Das, head of the Department of Microbiology and Joint Coordinator, IQAC, to kindly inaugurate the session with his welcome address. Sir,
Uh,
over to you.
Thanks, Movani. I wish you to perform a marvelous job as the program anchor for this session. This is Dr. Prashanjit Das, Head, Department of Microbiology and Joint Internal Quality Assurance Cell Coordinator, Jyotish College. Due to some technical issues, our respected principal madam is unable to join this program. So, on behalf of the Department of Microbiology and Jyotish College as well, I first of
Dr.
all,
Das, we couldn't hear you.
sorry, thanks, Mohoni. I wish you to perform a marvelous job as the program anchor for this session. This is Dr. Prosenjit Das, Head Department of Microbiology and Joint Internal Quality Assurance Cell Coordinator, Jyotish College. Due to some technical issues, our respected principal madam is unable to join this program. So on behalf of the Department of Microbiology and Jyotish College as well, I first of all would like to convey my heartiest welcome to the speaker for this session, Dr. Ranodip Chakraborty, Professor, Department of Biotechnology, North Bengal University. As it was already mentioned in the flyer, his topic of today's talk is Sneaking SARS-CoV-2, Molecular Distinctiveness and Mutation Spectra. This talk will mainly focus on the origin aspect of SARS-CoV-2 in parity with the main theme of this webinar, that is in search of there are some. Shudip, can you please stop your audio? Shudip, that is in search of origin, prevention and possible cure for COVID-19. Dr. Chakraborty is an expert in the field of molecular microbiology and a renowned speaker. So I am sure that this is going to be an exciting session for all of us to listen and interact with the speaker. We have already organized a couple of webinars related to SARS-CoV-2. Still, we organized this webinar yet again because an unprecedented level of efforts is going on in the scientific community to know the molecular biology, epidemiology, immunology, and therapeutics related with this pandemic causing deadly virus, and many new reports are emerging very frequently. This session will divulge many such evolving aspects about SARS-CoV-2. I also welcome the participants coming from different educational institutions across the state and beyond who are here listening via YouTube live streaming. Please put your queries regarding speaker's talk in the message box. We will convey the messages to the speaker during the question answer part. Please do interact to make the session an exciting one. I would also like to say that it will be our privilege to welcome Dr. Chakraborty physically in our college when situation will become normal again. We all are hoping that it will be very soon. So now it's over to Mogoni to proceed further with the program. Thanks.
Thank you, sir. I would now like to request our respected Professor Dr. Shudip Shamaddar to introduce the speaker for session 1, Dr. Ranodip Chakraborty. Over to you, sir.
Thank you, Mogoni, and good afternoon to everybody present in the first session of international webinar on In Search of Origin, Prevention and Possible Cure for COVID-19, organized by the Department of Microbiology in collaboration with IQAC, Jyotish College. It is my privilege to introduce Dr. Ranodip Chakraborty, Professor, Department of Biotechnology, University of North Bengal, and the eminent speaker for this session. After obtaining his PhD from the University of Calcutta, Dr. Chakraborty joined the Department of Botany, University of North Bengal in 1996 as an assistant professor. Since 2004, he is attached with the Department of Biotechnology in the same university. He is an environmental microbiologist, and his works focus on molecular microbiology, microbial genetics, and microbial genomics related to environments. He has published more than 70 research articles and 10 book chapters from reputed international publishing houses. He also attended many international and national conferences as an invited speaker and chairperson for different scientific sessions. Professor Chakraborty mentored a number of doctoral and postdoctoral students to complete their research works, and his mentorship is still going on. In 2019, Professor Chakraborty was awarded the Best Professor in Biotechnology Studies by Diwang Mehta National Education Awards. We are very much grateful to him for compromising his busy schedule and sharing his scientific expertise with us in the perspective of present situation. On behalf of the organizers, I welcome Professor Chakraborty in this webinar and request him to deliver his lecture entitled Sneaking SARS-CoV-2 Molecular Distinctiveness and Mutation Spectra, which will definitely open a new path to understand this pandemic virus. Thank you, sir. And over to Mogoni to continue the session.
Thank you, sir. I would now humbly request our honorable speaker, Dr. Lonodhi Chakraborty, to kindly deliver his lecture on today's topic. Sir, all yours.
Thank you. Thank you, everybody, and a very good afternoon. First of all, my thanks goes to the organizers. Organizers of Victory Fort, Vijayagar, Jyotish Ray College. And also a special thank to Dr. Das, Prasenjit Das, who is the present head as well as IQAC secretary and other faculty members. As I've just have a quick look in your department, started in 2000, now it is 2020, in 2005, you've started post-graduation. In 2017, you've got the boost. And I've seen that this department is also pursuing very good amount of research. So with all goodness, let me start my talk today. So can I share the screen now?
Yes, sir.
Now you need to, on this,
Put,
screen sharing.
yes, present now. You have to select the present now button.
Where is it?
It is below right of Google Meet
The
screen.
right hand corner below.
Please, Shilajit, help, sir.
Yeah, I got it.
Sir,
I got it. So entire screen. So should I?
you can select the PowerPoint also as a window. Select window. A window is there as an option. When you click a window, there will be the PowerPoint presentation. You just select it. Just. Thank you, sir. It's visible. Sir, I think you have presented the entire screen. Sir, you just maximize the PowerPoint now. It's in the down. Just maximize it.
It's in the down.
And in the down button. Just below. Not there, sir. Sir, in the navigation button in the bottom, there's a PowerPoint. Down, sir. In the navigation button, sir. Pan in the panel.
This one?
More left.
Is it?
The PPT.
The PPT.
The PPT below.
So it's reduced. Sir, you can see the Chrome sign, Google Chrome.
No, I cannot see it.
No. At the tab on Start button, beside Start button.
Beside the
Start button at the below.
There is a turn on caption, then you are presenting.
No. In your main screen.
In your main screen.
In the main screen, right?
In the main screen, down there's the PPT is reduced. Your PPT.
PPT in a minimized form there.
In a minimized form. Yeah.
This one.
Yes.
You make a full screen, sir. Could you?
Yes. Fine.
It's okay. Fine. Thank you, sir.
Okay. So after a small technical glitch, let me come back to the main talk. And you have already been exposed to what is the title. May I not just repeat this? When you look at the screen, it's a beautiful picture. a picture drawn by an artist, and if you can identify, the lady is opening a box, and the box is the Pandora box. And this Pandora box, this goes back to the mythology. We have Hesiod, a great Greek poet who is contemporary to Homer, has given the story of this Pandora box. And that, if you trace back that particular great poetry, you find Prometheus stealing fire from the god, and the goddess, the king of the god Zeus, was so angry he wanted to punish human beings. So therefore, this lady was made and sent to earth as a wife, as the wife of the brother of Prometheus, Epimetheus. Zeus said, while presenting this gift to the lady, Pandora said, don't open this box. And this box is the Pandora box, which the Greek myth has comes to the mind. The god has given the Pandora a locked jar, but the lady, driven by the human weaknesses, she nevertheless opened it and released the world misfortunes and plagues. So with that, I'm going to describe the perspective of SARS-CoV-2. Today, 18th July, if you recall, the total number of cases affected, you'll be finding that India rose to the 3rd position and Russia came to the 4th. So 3rd and 4th, so it was a supersession. And from where the China, this particular SARS-CoV-2 was discovered and spread, China came down to the position 26th. So if this is the perspective that it is fastly affecting the most populous countries of the world, and it has already affected 213 countries and other territories to a total extent of 215. So entire globe has been affected. The nature of this is so big, we call it pandemic. And we must realize that in our crowded world of 8 billion people, and when we have a combination of altered human behaviors, environmental changes, coupled to inadequate global public health mechanism, and this has all made, these animal viruses into existential threats. But we must not forget, that we have been watching this drama for more than a millennium. In case of pandemic influenza, which began with the virus of wild waterfowl, which has switched to the humans and then caused human-to-human transmission. So a bird virus became a human virus. And corona emergence takes a different trajectory. But the principles are more or less similar. Today's talk, the USP of my talk today will be a question that have always pierced in your mind, that how this SARS-CoV-2, what's behind its pathogenicity? We understand how it enters into the cell. We understand its spread and transmission mechanism, but we are very much worried about the sneaky behavior of its pathogenicity. So that part will be one of the important aspects that I will be dealing in details. Now, that's what I talked about, the pathogenicity of the novel coronavirus. Coronavirus encoded many accessory proteins which may be playing critical roles in virus-host interaction. But the fact remains that the function of this COV2 encoded accessory proteins are not well understood. So if you look at the time span or the life of this particular virus, talking about December 19, 2019, and now July 2020, near about 8 months period time, the knowledge, the scientific knowledge, it is just coming up. And interestingly, coronavirus, particularly this novel coronavirus, it induces apoptosis. The body system uses programmed cell death to do away with the virus. In most of the cases, the body wants to do away with the invading virus by switching apoptosis. But interestingly, this virus is the reverse one who induces, apoptosis. So there is a big question whether SARS-CoV-2 or its function now, with this background of pathogenicity, which we'll be talking in details, we must have something in parallel with the history. So my lecture will be to some extent blended with the historiography of pandemics. You are now viewing, really, a picture of influenza outbreak in 1990. And this is the picture of United States of America. And in the successive slide, you'll be observing the Kansas Army barrack of US from where the influenza had spread. So here, you'll be finding the American soldiers in 1918 pandemic being treated. They're isolated, right? And interestingly, in 1919, there was a reprint of a poetry written by Kathleen O'Mara. Let me not debate about the origin of this poetry, but whatever the poetry being written so many years back, it resonates with the present condition throughout the globe. And people stayed at home and read books and listened, and they rested and did exercises and made art and played and learned many new ways of being and stopped and listened more deeply. Someone meditated, someone prayed, someone met their shadow, and people began to think differently, and people healed. And in the absence of people who lived in ignorant ways, dangerous, meaningless, and heartless, the earth also began to heal. And when the danger ended and the people found themselves, they grieved for the dead and made new choices and dreamed new visions and created new ways of living and completely healed the earth just as they were healed. There are so many Bengali audiences today here. I'm sure they are participating in this webinar. Let me recall about one of the greatest Bengali storyteller, Kotha Shilpi, Sharat Chandra Chattopadhyay. He first used the word quarantine. শ্রীকান্ত উপন্যাসের দ্বিতীয় খণ্ডে চতুর্থ পরিচ্ছেদে তিনি লিখছেন, পরদিন বেলা 11টা-12টার মধ্যে জাহাজ রেঙ্গুনে পৌঁছবে। কিন্তু ভোর না হতে হতেই সমস্ত লোকেদের চোখে মুখে একটা ভয়ে চাঞ্চল্যের চিহ্ন দেখায় দিল। চারিদিক হইতে একটা অস্ফুট শব্দ কানে আসিতে লাগিল, ক্যারান্টিন। খবর নিয়ে জানিলাম কথাটা ক্যারান্টিন কোয়ারেন্টিন, তখন প্লেগের ভয়ে বার্মা গভর্নমেন্ট অত্যন্ত সাবধান। Mind it, this is the condition of 1918, so it was not a plague, but it's an influenza flu pandemic. শহর হইতে 8-10 মাইল দূরে একটা চড়ায় কাঁটাতারের বেড়া দিয়ে খানিকটা স্থান ঘিরিয়া লইয়া অনেকগুলো কুড়ে ঘর তৈয়ার করা হইয়াছে। ইয়ার মধ্যে সমস্ত ডেকের যাত্রীদের নির্বিচারে নামাইয়া দেওয়া হয়। 10 দিন বাস করার পরে তবে ইহারা শহরে প্রবেশ করে। So this goes just a translation of it. The word quarantine was probably first used by Sarat Chandra Chattopadhyay, the 4th chapter of the 2nd volume of novel Srikanta, and I'm sure I need not to read this. So if you look at this Burmese army, and this is the British Burmese army, and which the deck and the ship that was talked of by Sharat Chandra 1918, by this time has visited Rangoon, right? So many people were affected throughout Asia, and because of the fact that the troops of World War I were deployed by these, they were transported to their own home countries. Japanese school children, if you look at them, during 1918 pandemic, they're wearing the mask of the present day women and children whom we are now accustomed with. But what happened to Mahatma Gandhi? In 1918, he was there in the Sabarmati ashram and he contracted Spanish flu. Mind it, the Spanish flu, this is a misnomer. Actually, that started from United States, but all the big of the countries who went to the war, right, they censored the data of death. Only one country where the press was free, that was Spain, and therefore the number of death that was reported was coming from Spain, and hence the word called Spanish flu came into the picture. So Gandhi was 48 years old then. And 4 years after that, he had returned to Sabarmati from South Africa. He rested, stuck to a liquid diet during this protracted and lifelong illness of his life. When news of his illness spread, a local newspaper wrote, Gandhi's life does not belong to him. It belongs to India. So this deadly flu, like today, has also, inflicted Bombay, which is now we call Mumbai, in June 1918. And the influenza flu, it has taken more than 17 to 18 million Indians. 1 million means 10 lakhs. So 170 means 1.7 crore people were dead, and which was more than all the casualties in the World War I. And India then bore a considerable burden of its death. It lost 6% of its people. So this picture gives you the picture of early July in 1980, where 230 people were dying, then every day, up nearly 3 times from the end of June. And the symptoms are like that of the COVID-19 today. The workers stayed away from the offices and factories. More Indian adults and children were infected than resident Europeans. The main remedy at that time, the Times of India wrote, is to go to bed and not worry. People were reminded of this disease, that it spreads mainly through human contact by means of infected secretion from nose and mouth. Exactly the situation 100 years after is remaining the same. The colonial authorities, the British administration, they differed over the source of infection. The official Tarna, who was the health official, he believed that the people on the docked ship has brought the fever to Bombay. But the government insisted that the crew had caught the flu in the city itself. So you see, the blame game was there. And they said that it is due to the insanitary condition of the Indians was responsible for this great spread of this disease. Interesting, this is the picture which will speak about the colonial authorities also paid the price for the long indifference to this indigenous health problem, which is still existing today, since they were absolutely unequipped to deal with the disaster. Who writes? Miss Spiney, right? Laura Spiney, who is the author of The Pale Rider. This gives you a picture of epidemiology we just published in 2019. The spatiotemporal dynamics of 1918 influenza pandemic in British India. You will see that this epidemiology in history, this was accepted in September. Sorry, I made a mistake. It is not 2019, exactly 100 years after the pandemic. It's a famous paper that came out in American Journal of Epidemiology. Now you have seen what I said about the pandemic flu 100 years back, and what about today's data? Last night when I was surfing, I found Maharashtra death, and Bombay is the core of Maharashtra. And the death as on 17/7 is the highest number of deaths that is occurring in Maharashtra. And if you look at the Sabarmati Ashram, I have referred to that place, which is in Gujarat. It comes to the position, if you look at the death rate and the infected person and high. India during pandemic flu in 1918, and if you compare this with India during pandemic COVID-19 in 2020, to be sure the medical realities are vastly different now. Although there is still no cure, now we have the scientist who has mapped the genetic material of the coronavirus, which was not being done at that period of time when influenza flu has taken the toll. And there's a promise now of antiviral drugs and a vaccine. The 1918 flu happened in the pre-antibiotic era. But this is now happening in the post-antibiotic era. And there was simply not enough medical equipment then to provide to the critical ill. But we have the mechanism, but we cannot afford to give so many of the affected persons. So the health mechanism which a Rashtra, which the government should provide to its citizen, is not at all in a state of sufficiency. So all Western medicines were not widely accepted in India at that time. Most people, they relied on indigenous medication, but the things have changed. Now many people in India shall be accepting the Western medicines. Yet, there appear to be some striking similarities between these two pandemics, which is separated by a century. And possibly there are some relevant lessons to learn from the flu, right? See, the entire health department around the world are struggling to test for coronaviruses. So test, test, and test. That's the buzzword from WHO has a big meaning. More number of tests means more number of COVID-affected person. Whether or not their symptoms have come out or not, you can have an access to that number, and then only you can have a true epidemiological data. But by this time, the scientists have gone sleepless nights to come out with a vaccine. Coronavirus vaccines. But still then, there are 5 key questions that are even popping in the minds of the participant today. The first in human trial get going. In India, also the trial has gone. but key questions about how our immune system fights off the virus and how to safely trigger a similar immune response with a vaccine remain unanswered. So let me put you the 5 key questions. Do people develop immunity? If humans do develop immunity, how long does it last? What kind of immune response should vaccine developers look for? How do we know if a vaccine is likely to work? Will it be safe? So these 5 questions, you can have a look that these 5 questions put by Nature 579, the volume. These 5 questions has to be answered. So 1918 influenza pandemic, which was the deadliest one in the history, which took 50 million people's lives, equivalent to 5 crores or more death. And if you have the present population of the world today, extrapolate it to nearly about 20 crore people is going to die. And from December 2019 to July 18, means today morning, when I was updating the number of the death, number is 0.6 million deaths. And this is the rank of India reported on 1st July. So you look, India was in the 4th position, China in the 22nd position, and today is 18th. See the characteristic change. India rushed to the 3rd position, China slipped down to the 26th position. Read 26, 25th is Indonesia. So we must have to have some idea about its deadliness, okay? So that can only be measured that if we have an idea about the reproductive number for the 1918 flu. Now let me just tell you what is reproductive number. One person who is infected with COVID, when he infects some other healthy people, then how many number of healthy people become infected from an infected person? That gives you the reproductive number. In 1918, this I will discuss in details, okay? And it was around 1.8. And the reproductive number of this COVID-19 is now swinging between 2 and 2.5. And if this is so, then it is estimated that between 20 and 60% of the global population will ultimately be infected. And unless this 20 and 60, between these 2 percentages of the global populations gets infected, there's no chance of even imagining the herd immunity. I'll come to this point later. So this R0 or R0 is a very, very important index. What is R0? It is an average or expected number of people that an infected person could spread the virus to in her infected period. So if this R0 is greater than 1, definitely it is going to create an epidemic. And now it is more than 1, so it is pandemic. If this R0 is less than 1, then the outbreak will become extinct. If this R0 is 1, that is, 1 infected person will infect exactly 1 other person. And so the number of infected person in the population will remain constant over time. So we are here, R0 greater than 1. We will be striving towards a time where R0 gets lesser than 1. Now you need to have a fantastic epidemiological study. So you look at this particular reference when early in 2019, Hong Kong group of clinicians and scientists, they provided the first concrete evidence from human to human transmission. And this, I tell you, this is the excellent example of high quality clinical study, which can make a difference in policy setting. Several important clinical features of COVID-19 have been documented from this study. What are those? First, you see, and it was done within a family. Mind it, the spectra of this work is a small one, but its revelations are tremendous. So first, an attack of 83% within a family context. This is alarmingly high. And that indicates a high transmission rate of SARS-CoV-2. Secondly, the clinical manifestation, it ranges from mild, moderate, and serious. So mild to moderate with more systematic symptoms and more severe radiological abnormalities, which we see now. It is not restricted to the older patients, but also the mid-old and also the earlier ones. Third, that revelation said that there was a child who was playing within the family, did not show any COVID-19 symptom, was an asymptomatic child. But interestingly, when it was spitting SARS-CoV-2 RNA, and this is giving the first evidence of shedding from the asymptomatic virus and the possibility of transmission of SARS-CoV. Finally, the last was more interesting and very pervading and disturbing, that there were 2 young adults who were suffering from diarrhea, and which suggested the possibility of gastrointestinal involvement in SARS-CoV-2 infection. And therefore, it was showing a sign of fecal oral transmission. I will try to have these answers gradually. But if I just roll down the history of animal viruses, and if you look at these animal viruses, they are the only living entities, if you talk about, whose genetic material is high. So it is highly error-prone RNA viruses which is doing this disturbance because of its high mutation. Human species took 8 million years. It's 80 lakh years to evolve by one person. But there are many animal viruses which can evolve by more than one person in a matter of days. I'll take up this issue in the later part of my lecture. So it is not difficult to understand why we increasingly see the emergence of zoonotic viruses. So the virus is now going to switch the host. Now, what is the basis of switching the host when we talk about the SARS-CoV virus? If you look at this picture, you'll find everybody is now aware about what it is. So it looks like that. The brown buttons are the S proteins, which are the basis of getting recepted by the animal cell. So if you talk about the human cells, and if you talk about the pulmonary infection by the COVID-19, you'll be finding that human epithelial airway cells expresses a particular receptor to a very high extent. What's that? It is ACE2. ACE2. Now let me remind the listeners, there are 2 ACE. So ACE is an enzyme which acts as a vasoconstrictor. It's an enzyme that changes angiotensin 1 to angiotensin 2. And when it does so, it contracts the vessels. So it's a vasoconstrictor. So therefore, there must be another enzyme to counter vasoconstriction. So there must be a fine balance between the constriction and dilation. So vasodilator function is being imparted by ACE2. What it does, it actually hydrolyzes angiotensin 2 to make it a vasodilator. I'll come to this point. Now, if you look at this picture, the patient number 2, this is the photograph from Wuhan Hospital, which was published on day 8 and 11 after the onset of the illness. If you look at both the pictures on the second, on 8th day A and B photograph of 11th day, you cannot really see the lungs. It is fluffy. There is no degree of transmission of light. It's opaque, which is present in both images. So doctors of China, the Chinese doctors, they started their investigation by taking out the lavage from these pulmonary tract. And when they visualized this for the first time they observed, it looks like a virus and which resembles the earlier SARS-CoV. So SARS virus, it looks like SARS-CoV virus, but it was not established by that time that this is this Wuhan patient is infected by a novel coronavirus. When you say novel, because it has departed from the other, that is the earlier SARS-CoV. So something is there which has contributed to this newness or novelty. What's that? So how did they do it? They have to culture that virus, and they do it in cell culture. Human epithelial airways cell culture setup was done, from where the entire genome could be extracted, sequenced, and published. And after publication, we find that there is a remarkable departure from any other SARS reported earlier. And it is now we call novel coronavirus. And this is the timeline of the onset of coronavirus. From day 0, you might not see any symptom, but from the 5th day onwards, there could be the symptomatics here, and they can start. And when there is a shortness of breath, therefore you are put into the ICU or intensive care. So from 0 to 11, that was what the timeline of the coronavirus onset. See, before corona infected the globe, there were many other viruses which infected, but the extent of pandemicity is totally different. But if you look at the fatality of the other viruses, which is very, very high, that means number of persons who are infected with Ebola, right? The fatality rate can range to 50% of them would die. Then if you look at the mass, it is within 30 to 40. And when you look at the basic reproduction number that I dealt with some minutes before, what was that? This RO is a little bit higher than the seasonal flu or influenza flu or 1918 pandemic flu. See, the brown dot, it is lying within 2 to 3, right? But its fatality is very less if you speak about the others. Like SARS also, the fatality rate is 10%. But in case of COVID-19, it is not that fatal, but the rate of infection, the transmissibility is tremendous. We'll talk about it. So this is the point of focus. That is, if you really want to thwart its transmissibility, you have to do something at the entry point. And if you really want to do something with the entry point, you need to understand the peculiarities, what it has in its spike protein. Interestingly, this SARS-CoV virus, which we just talked about, it has a receptor, and I talked a little about ACE2 receptor, which is nearly present in most of our organs. So it gives a cue that SARS-CoV-2 will change its tropism. So it will show tropism. Its tropic activity is going to occur. It may not stay in your lungs. It might go to your intestine, duodenum, because these cells of duodenum, small intestine, lungs, and other organs, I will show you later on, it expresses this particular receptor, which we now know as ACE2 receptor. And this, I may not spend a little more time here because already I talked about what angiotensin converting enzyme is. And what is the function of ACE2? I have given you a brief idea that ACE2 is counterbalancing ACE because it cleaves the carboxy terminal amino acid, okay, N-terminal amino acid phenylalanine from the angiotensin 2, and this leads to a vasodilating activity. It contributes to vasodilating. But one very important question that might be popping up in your mind, that what else besides, vasodilation function? Do ACE2 regulate some other thing? Yeah, it regulates membrane trafficking of the neutral amino acids transporter. So neutral amino acid transported within the cell. So it is a very, very important protein which is expressed on the surface. And that surface protein is now the target of such fibers. And when the location of ACE2 is concerned, it is present almost in most of the organs, starting from the small intestine to the venous endothelial cells. When you talk, this is very dangerous thing. When it is going to infect your blood vessels, the interface between the blood and the blood vessel, there is high expression of ACE2. And therefore, the virus can infect those endothelial cells, causing endothelitis. So there will be tremendous amount of edema formation, and the whole blood flow within the vessels will narrow down, and that will strike something else which is going to hit your brain. So therefore, you will have several cerebral attacks. And it can migrate to your brain cells. So brain cells also expresses ACE2. So therefore, if you talk about the tropism of SARS-CoV-2, it can well move from one organ to another. So.
Dr. Chakravarti somehow got disconnected, I guess. So please bear with us. We are going to contact him.
Hello, Prosenjit.
Yes. Yes, I'm trying to contact him. I'm trying to contact him. Please wait for some few minutes. Okay. হ্যাঁ, আমি প্রসেনজিৎ দাস বলছি বিজয়গড় কলেজ থেকে। না, কোনোভাবে আপনি ডিসকানেক্টেড হয়ে গেছেন। হ্যাঁ আমি ওখানে কন্ট্যাক্ট করলাম, মানে আপনি কোনোভাবে ডিসকানেক্টেড হয়ে গেছে। হুম 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 ঠিক আছে। না না, আমরা ওয়েট করছি, আপনি দেখুন। হ্যাঁ হ্যাঁ হ্যাঁ, সেশন 1 বলে দেখুন, লিঙ্কটা দেওয়া আছে। হ্যাঁ, নাও ইউ আর কানেক্টেড।
আচ্ছা, আবার সেই এবার আমি আবার তো তোমার আপনার ইয়েতে ঢুকতে হবে, ওই পাওয়ার
হ্যাঁ
পয়েন্টটা
হ্যাঁ,
কোথায়
পাওয়ার
আছে?
পয়েন্টে ঢুকতে লাগে।
পাওয়ার পয়েন্টটা, hello, please help me to get into my PowerPoint.
শিলাজিৎ,
ফুল স্ক্রিনে গেলাম, শিলাজিৎ বাবুকে একটু বলুন।
হ্যালো শিলাজিৎ।
হ্যালো হ্যালো।
হ্যাঁ স্যারকে একটু হেল্প কর।
হ্যাঁ হ্যাঁ হ্যাঁ
হ্যাঁ বলুন তো।
স্যার একটুখানি ওই প্রেজেন্ট নাওতে গিয়ে ফুল স্ক্রিনটায় যান। প্রেজেন্ট নাওতে গিয়ে ফুল স্ক্রিনটা ক্লিক করুন।
আচ্ছা, প্রেজেন্ট নাওটা কোন জায়গাটায় আছে?
ডান স্ক্রিনের ডান দিকের নিচে,
ডান দিকে,
এই টার্ন অন ক্যাপশনস এর পাশে।
প্রেজেন্ট নাওতে না?
হ্যাঁ,
ওখানে কি কোথায় যাব? উইন্ডো?
হ্যাঁ হ্যাঁ একটা উইন্ডোতে যান, উইন্ডোটা যান।
আচ্ছা
উইন্ডোতে গিয়ে ওই পাওয়ার পয়েন্টটা সিলেক্ট করুন যেটা আপনি প্রেজেন্ট করছিলেন।
আচ্ছা, আবার সেই সমস্যাটা হলো। দেখা যাচ্ছে
না স্যার দেখা যাচ্ছে না।
আচ্ছা
স্যার আপনি তাহলে ওই ফুল স্ক্রিনটাই করুন, আমি গাইড করে দিচ্ছি আপনাকে। ফুল স্ক্রিনটা করুন।
আচ্ছা, জাস্ট এখানে আছে ফুল স্ক্রিন। ফুল স্ক্রিনটা
হ্যাঁ
এটাকে
লেফট ক্লিক
করলাম।
প্রেজেন্ট তো কিছু হচ্ছে না স্যার, সেই জন্য।
আসলে ইয়াহু প্ল্যাটফর্মটা, আরে গুগল প্ল্যাটফর্মটাতে আমি অভ্যস্ত।
স্যার এখানে কোথাও দেখাচ্ছে কি শেয়ার, মানে শেয়ারিং স্ক্রিন বা স্টপ শেয়ারিং এরকম কিছু অপশন আসছে?
কিছু নেই। এক্সিট ফুল স্ক্রিন, টার্ন অন ক্যাপশন, ইউজার ফোন।
আরেক ওই স্যার একটা একটু রিকোয়েস্ট করবো স্যার, আপনার প্রেজেন্টেশনটা একটুখানি পাঠাতে পারবেন, তাহলে আমরা এখান থেকে প্লে করতে পারি। তাহলে
হ্যাঁ নিশ্চয়। সেটা করা যাবে?
হ্যাঁ হ্যাঁ, শিওরলি শিওরলি স্যার। প্রসেনজিৎ দা, হ্যালো।
হ্যাঁ, আমার মেল আইডিতে স্যার একটু পাঠিয়ে দিন।
ঠিক আছে। মানে
হ্যাঁ স্যার, ঠিক আছে। এবারে স্যার একটুখানি ওই নিচে যে পাওয়ার পয়েন্টটা কোথায় স্যার? পাওয়ার পয়েন্টটা একটু ওপেন করুন, আপনার পাওয়ার পয়েন্টটা ওপেন করুন। হ্যাঁ, এবার ফুল স্ক্রিন করে দিন, ব্যাস তাহলেই হবে। আর আপনি আনমিউট হয়ে আছেন স্যার। ফুল স্ক্রিন। হ্যাঁ, আপনি আনমিউট হয়ে আছেন স্যার। আনমিউট। স্যার ঠিক ছিল এর আগে, আপনি মনে হয় স্টপ প্রেজেন্টিং করে দিয়েছেন। যেরকম ভাবে করছিলেন ওরকম ভাবেই করুন, তাহলেই
ঠিক
হবে।
আছে, ঠিক আছে, ওকে। নাও ইউ আর গেটিং?
হ্যাঁ, আপনি পাচ্ছি স্যার।
আচ্ছা, আমি কোথায় ছিলাম বলুন তো একটুখানি। আচ্ছা, স্লাইড গুলো দেখতে থাকুন এবং বলুন যে এই জায়গাটা অবধি লাস্ট আমি বলেছি, তারপরে আটকে গেছে, মানে ডিসকানেক্টেড হয়েছে। বিকজ আই ওয়াজ ইন আ ফ্লো, আমি তো বুঝতে পারিনি। আর আমার এটাকে অন অফ করে রেখেছিলাম। হ্যালো, ইউ আর হিয়ারিং মি?
ইয়া স্যার, এইখান থেকে স্যার, এখান থেকে।
এইখান থেকে পুরো অফ ছিল? আর ইউ শিওর?
ইয়েস স্যার।
No, sir. From location of S2 within
AC2।
হ্যাঁ স্যার, হ্যাঁ
হ্যাঁ, এই তো অনেক পিছনে চলে গেছে। এটা হ্যাভ ইউ সিন দিস?
হ্যাঁ, ইয়েস
দিস ওয়ান?
এসি, হ্যাঁ
দেন টাইমলাইন,
এই টাইমলাইনটা ছিল, হ্যাঁ।
this I was explaining, this one,
ইয়া
this one.
হ্যাঁ, এইখানেই আপনি ফাংশন, এইখানে ছিল।
Okay. So let me just call back. Sorry for all the participants who were there. So I was talking about the furin protein. Furin is another enzyme which is present in human beings, which plays an important role in cleaving the spike protein in a position so that the spike protein now becomes activated. So it always stays in a lying state, and it only stands up during binding with the ACE2, right? See how it does. This furin cleavage site is so important, which is not present in other beta coronaviruses. Do not contain this cleavage site, and this is one of the wonderful, adaptation of the SARS-CoV-2's protein. So how this ACE2 furin and the SARS-CoV-2 spike, both of them, all these three, they contribute to the spike's interaction with SARS-CoV spike's interaction with ACE2. And the coronavirus entry point is the most important virus entry point where people look at in order to find certain solution to prevent SARS-CoV-2. All of you, are you hearing me, all of you, please?
Yes, sir.
Yeah. So if you look at this picture, this gives you a fantastic one-liner what actually the furin protease do. Look at the A part of it. When the virus releasing out from an infected cell wants to infect another cell, it binds only when a cell surface proteases cleaves it. So if you look at down at B, you will be finding what really happens at the molecular level. At the molecular level, the RBD, which is this is receptor binding domain, it stands up. And when it stands up, the affinity gets more, and therefore the entry into the cell becomes much better. And this will become more easier to understand when you look at the RBD lying down for the immune evasion. So when it is pre-activated by furin protease I talked of, which is one of the addition for SARS-CoV-2, and when it goes to the standing site, right? So it immediately binds at higher affinity with ACE2. So human ACE2 here, so there is a protein-protein interaction is much, much higher in a standing state. And that gives you, the B panel gives you what is the difference between earlier SARS and this novel SARS-CoV-2. So when we quickly recapitulate, we find that RBD, they constantly switch between a standing up position for a receptor binding and a lying down position for immune evasion. So moreover, in order to fuse this membrane, the SARS-CoV spike requires to be cleaved at S1, S2 boundary. And that undergoes a dramatic structural change. And this SARS-CoV entry activating proteases, this includes a lot of cell surface proteases and lysosomal proteases. The information that I have shown has been derived from this famous paper that was published in PNS in May 26, just 2 months back. Cell entry mechanism of SARS-CoV-2. Now the question comes, why it is so pathogenic? And I talked about certain accessory proteins, which has just been revealed that actually controls the pathogenicity. Earlier, when I started giving this lecture, I said that viruses are evaded by the human system by the mechanism of apoptosis. So cell death is purposefully done by the human cells in order to evade the viruses. But here, SARS-CoV itself, it induces apoptosis or programmed cell death. So there is a big question that SARS-CoV-2A, 3A, this is a particular protein, accessory protein whose function was not known until few days back also. What's that? You see, this is the cell death related consequences of viral infection. You'll find a clear-cut division between two types of, two divisions. One, reduction. That means viruses like herpes, pox, and adenovirus and baculoviruses, they reduce this apoptosis. So they can stay there for a long period of time within the cell. But interestingly, Ebola, HIV, adenovirus, and also the coronaviruses, they increases the cell death. So what these scientists have done, which we come to know that this particular 3A, this is a structural protein of SARS-CoV-2. I will deal in details. So this ORF3A, this particular protein, is one of the 9 structural proteins. And there are 15 non-structural proteins. And non-structural proteins constitute 2/3 of its own genome. So only 1/3 is contributing to the structural part of SARS-CoV-2. What do you mean by structural part? Means all these, membranes and other spikes, envelope, these are the structural part of it. And one of them is an ORF3A protein. Nobody knew what's the function of this ORF3A in SARS-CoV-2 infection. So what they did, they cloned it and expressed that particular protein. And when these cell lines, HEC2, HEPG2, and VERO-E6 cell lines, when you are infecting with ORF3A, what happens? The percentage of apoptotic cells, they get increased. So if you look at this figure, you will be finding the vector means the open vector which do not have the ORF3A, and the black one is the SARS-CoV-2 3A protein is expressed. The percentage of apoptosis is going towards 30%. And it is varying from one cell type to another cell line. So HEC, HEP, and VERO, they're more or less all these cell lines are showing an increased apoptosis. What these ORF3A do? They resemble like the death receptor. You see, this protein, these Caspase proteins, I will deal a little bit details for the postgraduate students. The Caspase is something which is going to induce apoptosis, programmed cell death. The cell is programmed to die. And there are receptors here, which we call as death receptors. Interestingly, ORF3A, which is expressed within the cell, human cells, they are going to locate in the membrane of the cells. And during this membrane location, it is going to cleave Caspase 8. Now, Caspase 8 is in a silent state. It does not induces apoptosis until it is cleaved and it is activated. I'll come to that. And once this Caspase 8 is activated, so there will be a truncation of bit. And this T bit is going to induce the efflux of, cytochrome C will come out from the mitochondria. And there will be successively, there are a lot of changes that is going to happen. Now, this particular slide, please compare the left panel with the right panel. In the right panel, it is the earlier SARS-CoV. So the earlier SARS-CoV ORF3A can be in both the position, cytoplasmic localization as well as it is within the membrane impregnate. So it has the two part of it. But if you look at the left panel, you'll be finding the cytoplasmic part do not do any job. So when it is fixed with the membrane and a part of it lies in the cytoplasm, it is going to activate the caspases. How it does. So this is the caspase activation. So if you look at the first big rod, there are pro-domains, and when it gets activated, you can find this, the red one is getting clubbed with the YOLO2 and 2p20 and 2p10, that becomes an active caspase. So if you look at this, this is classically a death cycle. So here is the pro-caspase 8, which is lying without any function. So if it gets activated, the pro-caspase will turn into active caspase 8, and that will turn on it is going to cleave bid into truncated bid, And this truncated bead will then induce the efflux of cytochrome C, and this cytochrome C is clubbed together as an apoptosome, and then the whole cell is going to die. This becomes more clear in this picture. Let's say if you talk about this active caspase, forget about this one. This you can replace it with ORF3A. So ORF3A of SARS-CoV-2, which is a recent revelation, right, that it is fixed to the membrane, and from the membrane fixation, it is going to induce apoptosis. So it has a pro-apoptotic role. So when it activates Caspase 8, and you see here the bead, it becomes truncated, and ultimately there is a release of cytochrome C. Cytochrome C then induces apoptosome, and this apoptosome then again activates Caspase 9, and active Caspase 9, 8, all together they bring the apoptosis, right? So this type of apoptotic cycle can occur in two different pathways, extrinsic and intrinsic. It can occur within the cell via BCL2, and it can be done outside by the cell by some certain triggers. And here, the virus's own protein acts as that death trigger. So now you understand the picture that the authors have done, and I was trying to relate all the backgrounds to give you a fullest picture of how SARS-CoV-OR3A does. So what is the principal finding? The principal finding is ORF3A of SARS-CoV-2, it induces the cleavage or it activates Caspase 8, whereas BCL2 expression levels were not affected. This has an implication. So it is extrinsic pathway. The cleavage or activation of this Caspase 8 is recognized as a hallmark of extrinsic apoptotic pathway, whereas BCL2 plays an important role in intrinsic pathway. And when these people have worked with ORF3 getting expressed, they found within the cells there is a level of truncated bead, cleaved Caspase 9, cytochrome C's level. They are all elevated in the presence of SARS-CoV-2 ORF3. And either when you're using an inhibitor to inhibit these caspases, this significantly suppresses SARS-CoV-2 ORF3A induced apoptosis. So all this, if you put together, it results that SARS-CoV-2 ORF3A can induce apoptosis via extrinsic pathway. And this is the seminal paper which was accepted in 8th June 2020. So you can just imagine, just 1 month back, the whole knowledge has come out about the unknown accessory protein of SARS-CoV-2, ORF3A. So ORF3A of SARS-CoV-2, it induces apoptosis in cells. And it was published in Cellular and Molecular Immunology paper. Now, this is where we are working at, right? Once we have the sequence of ORF3A, right, then we wanted to understand the type of mutation that ORF3A is getting affected with. Ladies and gentlemen, let me tell you the most affected mutated genes, point mutated genes of SARS-CoV-2 is ORF3 and spike proteins gene. So the genomic region which is responsible for production of ORF3A is highly affected with this type of mutations. So you can find out that any changes in ORF3A might even lead to the changes in the pathogenicity. So you can understand why I called it sneaking. Now gradually you are getting the juice of this particular lecture, that why it sneaks. Now, these scientists, let me go back, they have created certain mutations. Mutation of cysteine to serine in a position of 130 to 133 amino acid sequence. There are two C's, right? What they did, they changed the C to S. And they have also changed a tyrosine, which is in the 168th position of the amino acid sequence, to alanine, and they created these two mutants. So if they have created the mutant, then they wanted to see what happens when you are expressing this mutant in the cells. So let me just tell you, if you look at this, there is an underlying part of it, okay? So what we did, we tried to put all the amino acid sequences translated, from this, and we located this particular LWL CWKC. So these are two cysteine residues where the scientists have changed and have seen something. I will come to that. And there is another Y, this is the tyrosine, YNSB. This is another part of it which is very important for ORF3. Till now, the entire science is not known. But whatever the partially being known, that means that when scientists have mutated the C or this Y, they have changed marked differences in inducing apoptosis. What happens? See, the result is the immunofluorescence, the assay, they have found that the wild type ORF3A, right, they localized in the plasma membrane with punctuation in cytoplasmic staining. That means if you just remember the picture that I've shown, that ORF3A is locating in the membrane, a part of it is anchored to the membrane, the part of is within the cytoplasm. But when we have done the mutation here, C2S or Y2A, means tyrosine to alanine, they showed minimal apoptosis inducing. And the localization also changed. That means it came to the cytoplasm, not anchored with the membrane. So membrane anchorage is an important role to play in activating caspase. So both these mutants, they fail to induce the cleavage of bit, caspase 8 and caspase 9, or the release of the cytochrome C. So see, this is a foolproof experiment where they have even located the regions, right, which when mutated by a single amino acid substitution can change its pathogenicity. So these results indicate that membrane association is required for pro-apoptotic activity of SARS-CoV-3A. And all these results, they indicate that in SARS-CoV-2 ORF3A, the membrane association feature is involved, but not essential for the proactive apoptotic activity of SARS-CoV-3A, suggesting that two ORF3A proteins from different coronaviruses use different strategies to induce apoptosis. See the complexities. So therefore, if you remember, the title of my talk was molecular distinctiveness. So I'm talking about the molecular distinctiveness, right? And this is the part that is after 3,608 genomes that has been deposited, all of SARS-CoV-2 from different parts of the world, we created a pan-genome. So from that pan-genome, the entire SARS-CoV-3A, the mutations are mapped. The small red ones are the places or the nucleotide positions which has undergone transition. And the blue one are the transversions. So transitions and transversion throughout this particular ORF3A is inflicted with mutations. I'll come to that later. So the significance is, if you want to carve SARS-CoV-2, you need to understand how it enters the cell, And the second parameter got added today in the lecture is you need to understand the pathogenicity also. Now, if you just roll back to the human ACE2 and the SARS-CoV-2, that particular domain or motive, that is the receptor binding motive, you will be finding a striking divergence. Means the earlier SARS-CoV, the point of contact, and the present one, the point of contact is different. We are working on this aspect, that what happens to the structure function when there is a mutation that the CoV virus is carrying. So this is the entire transition and transversion mutations that has been already mapped in the spike. So this goes the entire mapping all across the spike, and the previous slide that you have seen that we have mapped all the mutation on the ORF3. Why we have mapped? I'll come to that answer later. So therefore, we now understood, this, any changes on this contact point can create certain havocs. So a key to tackling this pandemic is to understand this receptor recognition mechanism of the virus, which regulates its infectivity, pathogenesis, and host rates. And this, this is a seminal article again, which was published in Nature, 14th May 2020, the structural basis of receptor recognition by SARS-CoV-2. I was talking about the blood vessel endothelial cells, which expresses, ACE2. So if by any chance the SARS-CoV-2 infects endothelial cell, there will be endothelitis, and that will cause lot of vasoconstriction, artificially induced by edema, because all the cells, they will undergo a lot of inflammatory responses will take place. The whole part of it will swell and the blood cannot flow. There will be a large pressure build-up that is going to strike elsewhere. The vessels is going to be snapped. This is just a picture about the details of the endothelial cells, which is in direct contact with the blood cells. Now, this picture was an autopsy-related picture of a COVID patient who died. And after that, this was done in Zurich, in Switzerland. All the organs, they were dissected, and they found the presence of SARS-CoV-2 in all these organs. So that gave a cue to these scientists to understand that whether or not these organs which are showing the SARS-CoV-2 are really can accept SARS-CoV-2 infection or not. So they did it in the cell lines. So it has a panel of large number of cell lines were taken, starting from respiratory tract, gastrointestinal tract, liver, cervix, kidney, neuronal, means they have taken glioblastoma, which is also can receive SARS-CoV, and SARS-CoV-2 virus can infect glioblastoma cell line. Human muscles, okay? And they have also compared it with other animals' cell lines. They have used dog, cat, porcine, rabbit. This is a spectacular experiment. I'll come to that later on. So if you look at the full-length SARS-CoV and SARS-CoV-2 proteins in terms of the spike protein, you will find a degree of similarity and also a degree of dissimilarity with just the earlier SARS. So there is a tremendous diversion when SARS-CoV and SARS-CoV-2. And when you look at the phylogeny, you will be finding that entire human SARS-CoV strains, which we will be later on calling as quasi-species, it is clubbed with pangolin, and up to that it has a bat. So all these spikes, proteins, they have a lineage. That means SARS-CoV-2 spike has a lineage. Immediate lineage, you have the pangolin and bat. This is a region which corresponds to SARS-CoV, SARS-CoV-2. When you put all the amino acids one after another and align, you will be finding, if you look at this RBM, you'll find a gap here. And this will give you a clear picture, which I was trying to emphasize, the S1S2 cleavage site. The red arrow gives you the S1S2 cleavage. And if you look at SARS-CoV-2, none other coronaviruses, they bear this particular bridge. So there is a S2 cleavage site also. And when you look at the protein models of SARS-CoV, any person, it will be difficult for any specialized person, unspecialized person to demarcate what's the difference between these structures. But if you look at this SARS-CoV structure, CoV-2 structure, and this bat-CoV structure, they look alike. But there are differences which one needs to probe into the details. This is the expression profile of ACE2 in our different organs of human being, starting from duodenum, gallbladder, heart, kidney, small intestine, stomach, testes, thyroid. Where not? So all these organs can be, infected if there is a possibility of interaction between SARS-CoV-2 with these cells that are expressing ACE2. So naturally, we are driven to understand that whether or not the ACE2 across the animal kingdom and those animals we picked up which are domesticated by human beings. And strikingly, if you look here, it is a great clade, okay? So this big clade, which has a common ancestor, takes into account, this is our cow, this is buffalo, right? So all the animals which we are acquainted with are there. Camelus, which is a Somali camel, Suscrofa, which is a wild boar, horse is there, then capra, that is a domestic goat, sheep, cow, and the next one that you find here where human ACE2 directly forming the closest relationship of ACE2 of human beings with pan troglodytes, which is of chimpanzee, and this one for orangutan, and this one the normal rhesus monkey. So all monkey, orangutan, then chimpanzee, human being all clubbed together. When we look at the evolutionary history of ACE2, now we go into the little bit genomics part of it. I don't know whether, should I continue? Okay. So as I said earlier, that 2/3 are actually expressing the non-structural polyproteins. And 1/3 of it is the spike protein. The structural protein, amongst which this ORF3 is a component here. So if you look at the SARS-CoV complete genome, this is the Wuhan genome, which all across the world, we take this as a reference genome. Means upon which all the mutations were mapped. So therefore, whatever the divergence that took place in different population, we are going to map it on the Wuhan strain, right? And that is 29,903 nucleotides. It's a huge RNA genome, which you have 2 ends, that is 5' untranslated region. There is a 3' untranslated region and these 2 flanks all the genes. There are 16 NSPs or non-structural genes and 9 structural genes. And ORF3A is here within. See, after S, that is spike protein, you have ORF3A. It's a big protein. And another spectacular thing is, this is a positively stranded mRNA. What does that mean and what does that imply? It implies that as soon as it enters into the cell, it is capable of translating because it is as good as the mRNA. And you have a poly-A tail in the 3' end. So therefore, it takes advantage of the translation machinery of the human being or human cells to transcribe some of the very important proteins at the earlier phase. Okay? And this is done by a complex. What's that? Replication transcription complex. And that is actually this whole 16 genes, they are taking care of this replication transcription complex. Many of the accessory proteins here even within these NSPs are not totally understood. So therefore, this gives you a genetic mapping of all these positions of NSPs and other structural proteins later on. So you need to understand the architecture of this SARS-CoV-2 transcriptome. What is meant by transcriptome? In a time instant, what are the types of RNA that is generated? mRNA generated. You'll be surprised that there are subgenomic RNAs created, and from there, the subgenomic mRNAs are created. Let me make it clear. What is that? You see, if you want to have the mRNA, number of mRNAs, so the plus strand has to be changed to the negative strand first. And taking the negative strand as a template, you will have all the plus strand created. And that plus strand, unless the plus strand created, it will never be behaving like an mRNA. So all the mRNA is equivalent to the plus strands. And the genome itself is a plus strand. So if you want to have lot more plus strands, right, you have to have the plus strand converted to negative strand first, and on the negative strand, all the plus strands is going to be transcribed, and finally it has to be translated. So look at this. So this is this will give you the picture of canonical subgenomic RNAs that I talked about. Now, subgenomic RNAs and subgenomic mRNAs are different. First, you have to create the subgenomic RNAs, upon which the subgenomic mRNAs will be created. And look here, that gives you the schematic presentation of the canonical subgenomic mRNAs, all having poly-A tails, right? So, and therefore, these are all which can be acted upon by the translation machinery. So if we quickly recapitulate, then the genome of the SARS-CoV-2 is a positively stranded, positive single-stranded RNA, which has 2 caps. Genomic RNA will be used as template to directly translate this polyprotein 1A and 1B, which will be encoding the non-structural proteins to form replication transcription complex. And that will be put into a double membrane vesicle. And subsequent to that, there will be a nested set of subgenomic RNAs or sgRNAs will be synthesized by RTC in a manner of discontinuous transcription. So here lies again molecular distinctiveness of SARS-CoV-2. And these negatively stranded sgRNAs will then serve for templates for production of SG, means subgenomic mRNAs. Approximately, I told you, 2/3 of the whole genome encodes for 16 NSPs. And there is a -1 frame shift between ORF1A and 1B that leads to 2 polypeptides. And these polypeptides will be processed by virally encoded enzymes to form all independent proteins. So this is a quick recap of the types of function by all these NSPs that are being done. And this 12, NSP12 is that primer-dependent, RNA-dependent RNA polymerase. So this RNA-dependent RNA polymerase, it has to do a tremendous job for which its processivity has to be increased. And this processivity is increased with the help of 2 different proteins. And those 2 proteins are helping processivity, the NSP7 and 8. NSP8 is a primase which goes on feeding the small primers, upon which the entire RNA will be transcribed. So you can think about the molecular machinery of this small, tiny virus which is shaking the world. The world which has sent ships to Mars is incapable of handling a small 29903 RNA virus. So I'm just rushing to this. And this is when we take entire genome of this beta coronavirus SARS-CoV-2 with other viruses, beta coronavirus genomes, and when you do a phylogenetic analysis, you will be finding that Wuhan strain, this one, and the bat one is forming a clade. And you will have all other coronaviruses, phylogenetically, they are separated out. And when you look at this in a frond-like picture, you'll be finding the green frond has the SARS-CoV and SARS-CoV-2 markedly different from the other limbs. And finally, if you whatever I said, that after the entry, the host ribosome is going to play a role. There will be a translation of viral protein, and this RCT is going to create a lot of minus sense strands upon which the mRNA will be produced, proteins will be transcribed, and finally they will be packed, and it will go via exocytosis. And during this getting out, it damages the cell. The story that I have just revealed about the ORF3A. So it induces apoptosis. It breaks open, make it die, and that leads to a cascade of events which will be influencing others. So there will be a storm of cytokines. This is the structure of the RDRP. You have special structure, finger-like, palm-like, thumb. These are the different regions of RDRP. The people are working here to understand how these RDRP do, because this is, again, a very important and interesting part the scientists are trying to find out. Because if you have a drug which can prevent the polymerase activity, you are going to halt SARS-CoV. And therefore, remdesivir, which you have all known by this time, is nothing but a base analog. So what happens when it starts transcribing and when it takes up the base analog, it truncates the replication. So when there is a truncation of replication by this remdesivir, which is nothing but a base analog, which is pushed inside your body. So therefore, the promise for SARS-CoV-2 remedy lies within remdesivir-like drug, which targets RDRP. And I just said, NSP7 and 8, these 2 proteins are always associated with RDRP. So when it was crystallized and the whole crystal was studied, they found that NSP8 and NSP7 is an integral component of that crystal. Right? Now, the last part of my lecture is the part where I am interested with. So when we have studied the entire 3,608 viral genome sequence and created a pan-genome of SARS-CoV-2, then we are in a state of mapping all the mutation across that has happened within this 9 months' time. Interestingly, all the students, please, if you have studied classical genetics, you'll be finding that there are 2 types of point mutation: transition and transversion. There are only 4 possibilities of transition, right? C2U or C2T, T2C, then G2A, A2G. Purine, pyrimidine, pyrimidine transversions. But when we talk about transversion, there are 8 different, type of combination can occur. So naturally, if I pose this question to the participant, that what is the probability of mutation to be recruited in a genome, you can say that those who are having the greater access, that is 8 different combinations for transversion, will occur in a very high dimension. But nature has something different to say. Most of the transition mutations, they occur in the 3rd base of the genetic code. So if you have read the genetic code, you have understood that the 3rd base change, either it is a purine or a pyrimidine, actually it will code for the same amino acid. So that type of mutation is a silent one, which we call as synonymous mutation. But if the transition occurs in the 2nd or the 1st base of the codon, then it never remains synonymous. It becomes non-synonymous. Ladies and gentlemen, the transition after mapping, we have come to find out, the transitions, there are a lot of transition mutation that has occurred in the SARS-CoV genome. They are asynonymous or non-synonymous in nature, which in most cases the transversions do it. And interestingly, which is strikingly different, that is, we get a biasness, that is, large number of transition and lesser number of transversion. So there is a selective bias in SARS-CoV-2. This I spoke about. What is transition? What is transversion? And what are the different types of transition and transversion that can occur? And this also I explained about the synonymity or silencing of the mutation. If it occurs in the 3rd base, then if it is a purine or a pyrimidine, ultimately the amino acid remains the same. This was our starter. Means first, when these sequences were coming out, we took the sequences from 13 different places of the world and just used a blast. And that blast result has enabled us to enter into the more details. If you look at this table, you will be finding that amongst all the sequences that we used as a query sequences, the minimum matching only happened at 99 mismatching took place. That means, sorry, the reverse one, if you see the minimum identity that we observed only for one was 99.93. So all those populations of SARS-CoV-2 which was escaping from the patients throughout the world and got sequenced, they are just differing by decimal 0.1 to 0.7%, and that is creating a lot of changes. So we tried it over these 3, and then we tried to map these 13. But finally, you cannot stay here because already 3,608 genomes have come. At that time, we found there were spectacular absence when the SARS was just evolving, absence of mutation in the NSP7 and 8 and 9, which we did not find amongst 42, 21, and 46 Indian genomes, right? But gradually, when whole picture was opened, now you look at the genome spectra of the transition mutation. So we mapped all these 4 across the pan genome. And it will be strikingly, you'll be finding that gene S means the gene that is coding for spike is taking 107 mutations, right? And similarly, when we map transversion, we end up with, total of 660 transversion. And if I move to the previous one, okay. So now you got the essence. Now we tried to map the entire transition across the spike. And we selected one of the mutant. Now, which mutant, I'm not going to give you the details of it because this is the IPR part of our team, and which leads to, and when we are superimposing the Wuhan strain, that is non-muted one with the mutated one, we find a spectacular difference. Means the contact points remains the same, but the affinity differs. So therefore, the mutation in the spike can affect the binding affinity of the spike to ACE2. So any population having this particular mutation is going to do havoc in a population because the binding affinity of this mutated spike is higher than the wild type one. Again, let me go back to this. These are the contact points. And if I show you the previous, the contact points remains the same, but due to a mutation, which is not in this contact point, somewhere distant from there, which is controlling the entire structural property of this particular spike protein. And we tried to do the phylogeny of the SARS-CoV-2 quasi-mutants, means all these are the variants, okay? You will be finding that, the first China, that this one is the MN90893 is the China one, from where you have 2 distinct lineages which has diverged out. This is USA, and another strain of China is here, which is clubbing with Indian strain, and Iran, Japan together, South Korea, Brazil in a clade, and all having a common, ancestor. So ultimately, when this strain has escaped to the entire globe, there has been a polyphyletic evolution going on. If you remember, the amount of C2U conversion was tremendous. Why it is so? It is also not because of the randomness, because C2U conversion can occur even with the, outside the cell. The type of disinfectant that you are using, for example, UV, it spontaneously deaminates cytosine to uracil. And in this type of RNA viruses, when it enters with that deamination part, that is already the C has changed to U, then it is going to behave like a mutant one. And this mutation rate is not only, this is recruited, this is our hypothesis, okay, that excess use of disinfectants and others are also accelerating mutations of SARS-CoV-2. And here we have also pointed out one heat treatment with sodium sulfide and metabisulfide, which is used as disinfectant, antioxidant, and preservative agent. Use it at pH 7 and 65 degrees Celsius for keeping our foods intact. you'll be finding, and even the alkaline brines and marinades, which are being used in Chinese cuisines, okay, they can also change this cytosine. If you track this, this is a chemical changes that is taking place, okay? HSO3 is a sulfite, sodium metabisulfite is used, which changes, there is a changes to uracil. Similarly, there is a high intensity of A to G conversion by hydrolytic deamination. And also, when we searched the behavior of, cleaning behavior or sanitation behavior throughout the world, the Western world uses, dilute formic acid for cleaning. And this dilute formic acid can spontaneously change adenine to finally guanine. So these are the two chemical reactions that take place by human intervention to change the bases. And once the bases are changed and those are taken in, this takes up a new quasi-strain. This is my group. More collaborators are getting added to it. And before I end, right, excuse me, are you listening?
Yes, sir. We are listening.
Hello?
We are listening. Yes, sir.
Okay. So there is a small 1-minute video, and with that, I will end my lecture.
Sir, the video is not getting observed.
Is it?
Yes, we are not getting it.
Okay, I will run it again. See that you see it. Are you getting it?
No, sir. The icon is here, but the video is not getting observed.
Okay, nothing to worry. I end my lecture here with all thanks to everyone, right? I can read out actually what was there in the video. And with that word, I will finish. Imagine you were born in 1900 when you are 14, World War I begins. 22 million deaths has occurred. 1918, Spanish flu, 50 million people dead. And you're alive at that time. When you are 29, the global economic crisis took place. The collapse of the New York Stock Exchange, unemployment everywhere. When you are 33, there was Nazis coming to power. When you are 39, World War II begins. When you are 45 years old, 60 million dead, with the Holocaust of 6 million Jews died. When you are 52, Korean War took place. 64, Vietnam War begins, ends in 1975. A child born in 1983 thinks his grandfathers have no idea how difficult life is, but they survived several wars and catastrophes. Today we have all the comforts in our life amid a new pandemic, but we complain because we need to wear masks. We complain because we must stay confined to our homes. We have food today, we have electricity, Wi-Fi connectivity. None of that existed back in the day, but humanity survived those circumstances and never lost their joy of living. A small change in our perspective can generate miracles. We should be thankful that we are alive. We should do everything we need to protect. This message should reach everyone. Please help spread it. Thank you to all my listeners. Namaskar. Hello.
Thank you, sir, for your
Thank you.
extremely informative and insightful presentation. Now we shall move on to the question answer session. I'll request my senior Digongana Day, student from MSc 4th semester, Department of Microbiology, to conduct the session. Participants can drop their queries in the chat box on YouTube. Digongana Day, over to you.
Yeah.
Yes. Am I audible?
Yeah. I could hear you.
Thank you, sir, for giving
Thank
us
you.
such an informative and valuable lecture. This will definitely improve our knowledge regarding the molecular nature of SARS-CoV-2 virus. During your lecture, we got some questions from the participants, and I'm going to narrate one by one.
Yeah, you ask the question one by one, I'll answer it. Okay?
Okay, thank you, sir.
Yeah, first question, please.
Yes. Shubhajit Nath asked, is it possible to make a airborne cure for any virus by using nanotech?
Well, it's a probability, but you have to first imagine how you're going to kill the airborne ones. If you can, yeah, there is a possibility and there are mechanisms by which, but whatever air you take in, that has to pass through an electrode that is going to burn the particles. So if we have a mechanism of inhaling type of device which will take in air, and if the air contains SARS-CoV, that will be immediately, it will be cremated. That type of mechanism, if you can think of a device that will be working fantastic, rather than trying your hands on nano. Because I don't find, directly right at this moment that how you are going to pose nano or you're going to spray these nanoparticles in the air for killing. So if you want to stop the airborne, the inhalation part has to be checked, okay? So you can make a room free of CoV-2 by passing air through, for example, the ICU units or something that can be made, free. But there are a lot of innovations is required to bring out such machines. Thank you. Next one, please.
The next question is from Devoshmita Mundul. Sir, is there any chance to get infected by this virus more than one time?
Yeah, that is the question. And here lies the lot of, suspicions. Why suspicion? We don't know really that after a person getting infected, how long the antibody will be active. And there are reports that the persons are getting reinfected. And this reinfection is very dangerous. Even after vaccination, if the reinfection occurs, then that will be more dangerous. These are the questions that, if you remember, the first 5 questions that I put is yet to be answered. Next question, please. I have given my answer. So I believe
Yes,
I can, yeah, next question.
Yes. Next question is from Shobhik Roy. Sir, please can you highlight something on the correlation of a particular blood group and vulnerability to COVID-19 infection, as this information is floating in the air now?
Sorry, I cannot answer this question. I'm unable to answer this question because I do not have really any information right at this moment to answer this question, that whether the blood group and COVID infectivity, maybe there could be some relationship, but I do not have right at this moment to share with you. Next question, please.
Yes, next question is from Debayani Chakraborty. Can RNA interference technology be applied successfully in preparing a vector which obstructs specific mRNA coding, the spike proteins, so as to prevent infection and make a vaccine?
Yeah, people are trying, and this is on the way that, mRNA vaccines are being tried. That on expressing can give you some sort of, protection. So people are trying that way. Yeah, there lies a silver line. People are trying it. People are trying.
Okay. Can I go to the next question? Hello.
Hello.
Ananya Chakraborty.
Yeah.
Sir, can you please say about the success rate of current vaccine of ICMR that is going for trial?
This is one of the fundamental questions. Now, this can only be answered that, see the clinical trials which are going on. How do you know whether a vaccine will work? That is a big question. The vaccines go into the human trials after test of safety and effectiveness in animals. But vaccines being developed by the US drug firms like Moderna or Innova Pharmaceuticals, they are being tested in animals in parallel with human phase trials. I am not aware about the ICMR, but this Bharat Biotech one. Okay, the vaccinated animals will be infected with the virus to see whether they are protected. And as researchers learn more about the infection from human and animal studies, they will get a better sense of vaccines which are likely to work best. So therefore, the attempts, now you got my answer really, that in order to say that this will work, you have to pass through these stages of research. Next, please.
Shubham Vishwas, sir, even patients are recovering from this SARS-CoV, but there are no antiviral medicines, so how they are getting recovered?
Oh yeah, this is because of the low fatality rate. You see, like Ebola, I have shown you that 50% dies, but here people recover from it, and the recovery rate is also high because of the fact that it is not that fatal unless you are affected. Some other organs are also affected by COVID or other morbidity. All bringing together can bring down, that accelerates the survivability. That interferes with the survivability. But somehow, when these people are being treated, let's say a COVID-infected person showing some symptoms, going to a hospital, I am sure that the doctors are trying to stop that cytokine storm, right? And in order to stop that cytokine storm, there are drugs which are used in different other diseases, like, hydroxychloroquine. This gives you a platform to lower down your immune responses. It is in a low key. You get this answer from the bat. You see, the bat SARS do not kill the bat, because bat is an animal which is highly active, and the adaptive immune system takes longer period of time to react in bats, right? So instantaneously the inflammatory condition does not take place, and therefore they survive with the virus. So such a condition, if you create within a human being, which can happen genetically, means some of the persons, or all of us is a different, genetically different, and there will be difference in, reacting to the foreign substance. If reacting to the foreign substance, particularly the viruses, if it is low, it will take longer time to get a serious infection. By that time, some other drugs which are being used can help in revival from the COVID-19. So this can occur, this can happen, and we have to depend upon our innate immune system. I can even give a lecture 1 hour over this innate, because there are circumstances that melanin and vitamin D and some other vitamins which play an important role in, even they can prevent furin proteases. So there are systems which can, within our body, the body itself is a doctor. So you need to charge up those points. And if there are mechanisms to stop this furin protease, then the entire infection rate becomes low. So this can happen due to some other, because our knowledge about the entire body itself and the coordination, because just the system biology has opened up. So we know very little about how the systems and how the proteins react. So the entire system biology is in an embryonic state. Unless we understand everything, we cannot really answer your question. But there are instances that some other materials which are produced or taken by the human system can help in protection, preventing COVID, and also preventing the seriousness of the COVID. So therefore, many people, like Mahatma Gandhi also at that 1918 survived that terrible pandemic, influenza flu. Next question, please.
Shaimatika Dev. Do vaccine designing can be affected by the various changing mutations, because the strains are changing geographically due to mutations?
Yeah. That is why the vaccine designing becomes so difficult. So it may happen that you have evolved a vaccine for a strain which will work for India but may not work for, for any other Western world. So because of the rapid mutation getting recruited, the epitope will be changed. And if the epitope is changed, the vaccine will not work. So that is a possibility. So therefore, even within a population, suppose the vaccine has come, that may not stay stable for more than 6 months. This is a presumption. It can stay, but we don't know really this answer. So I started my lecture with the 5 big questions, which are yet to be answered, because we don't have the time to answer this question. No, it is not me to answer, but the entire globe to answer this question, because this virus is a pretty young virus and many things have to be experimented before we can answer this question. Yeah. Next question, please.
Next question, Shubham Barik. Sir, why vitamin D levels are low in COVID-19 patients?
Is it so? Yeah, it could be, because vitamin D is synthesized by the body via sunlight, isn't it? So sometimes genetically, in some places, lesser exposed within the hospital environment where sunlight is less, your exposure to the sun is less, gradually the body's own vitamin D level comes down. That could be one of the reasons why when you are staying indoor, the level of vitamin D is low. We have seen across the latitude, which is very interesting, that when you go up the latitude towards Sweden or something, and we have a fine relationship with vitamin D and melanin, and there is a relationship between vitamin D and melanin, and we have written a paper on that. Vitamin D and melanin can mitigate COVID-19. These are all hypotheses. We did not experiment, but all the circumstantial evidences. For example, Sri Lanka, if you look at the death of the Sri Lanka, is least because it is in the latitude which is near to the equator. So all the equator regions, countries are least affected because they have a high degree of vitamin D circulating in their blood. Okay, so there will be a relationship. We are yet to work it at that level, but we have the data on vitamin D content of the blood of different countries, and even there are very important studies we made regarding the migrated population. For example, Sri Lankan population which is migrated to Norway, and when the vitamin D was measured, it fall down. But when when those people were in, they are in, or majority of the population of Sri Lanka, there's a very high level of vitamin D. So the vitamin D synthesis is correlated with, latitude and also the sun length and everything. And even, in cold countries also, there are 6 months daylight, 6 months. Some of the countries have 6 months full length, day and night. And when they pass through a full night days, they have to take vitamin D in addition. Like food, which we have, they take vitamin D as a pills regularly. So this is something which is physiologically related with solar system, right? And the position, the geographical position, and nearness to the sun, and also the duration of the sun in a day. So all these contributes to the synthesis of vitamin D in the system. Thank you. Next.
Rajoshi Roy asked, how can lyotropic crystals be used to treat COVID-19?
Please repeat.
How can lyotropic crystals...
What's that lyotropic crystal? I don't know. Can the lady explain what is lyotropic, or if you can explain what is lyotropic? I don't have the answer, please. You can skip to the next question.
Next question is from Dr. Priyanka De.
Yeah.
We have heard that researchers' main safety concern is to avoid a phenomenon called disease enhancement. Can you enlighten on this aspect with SARS-CoV-2 vaccines?
Again, you have asked a question on vaccine. There are a lot of questions yet to be answered. What kind of immune response the vaccine developers look for? This is a very important question. A clinical trial that began last week, they focus on a vaccine developed by Moderna, a company based in Cambridge, Massachusetts. This vaccine consists of an RNA molecule that is designed to train the immune system to make antibodies that recognize and block the protein that the virus used to enter the human cells. However, a successful SARS-CoV-2 vaccine might also need to prompt the body to generate antibodies that block other viral proteins or make T cells that can kill infected cells. So this is how we can train and we can think of, but no concrete answers. Next question, please.
Sir, there are no more questions from the
Thank you. So there were a volley of questions, and all the questions who have raised a very intelligent question, except the one I couldn't answer, the lyotropic. I couldn't understand what's lyotropic. Maybe, you see, now so many people are coming up with so many jargons, which is impossible to track all these jargons. Well, all the listeners, you have been listening to a big, seminar which I have exhausted, and it was truncated in the middle. It got severed, and then again I tried to patch. So there was some sort of discontinuity also, which prevented me to give the 100%. But still, what is your impression? How the participants have got it? And may I have a quick response from the viewers or from you who were in the
Sir, from YouTube feedback that we are getting, your talk is excellent.
Thank you.
They are satisfied extremely. You have answered most of the questions and they acknowledge that. It's a very good session from the participants' viewpoint.
Okay. From the faculty members?
Definitely, we are overwhelmed.
Okay. Thank you so much.
Thank you, sir.
Thank you. So I can disconnect now, right?
No, sir. There is a small part attached with it. The vote of thanks for the question.
Please go ahead.
Okay, sir.
I'm in the line.
Thank you, sir, for clearing all our doubts in this wonderful way.
Thank you.
Thank you for this amazing session. Thank you, Digongana Di. Now I would request our respected Professor, Dr. Gaidi Shahakesh, Associate Professor, Department of Microbiology, to deliver the vote of thanks. Over to you now.
Thank you, Moboni. On behalf of the Department of Microbiology and Internal Quality Assurance Cell, Jyotish College, I would like to thank Dr. Ranodhir Chakraborty for his informative speech. Listening through his talk was an enriching experience. I would like to thank Dr. Rajoshi Niyogri, our respected Principal Madam, for her constant encouragement. My heartfelt thanks to all the participants who made this webinar successful. I would also like to thank my fellow colleagues of our college. It's my pleasure to thank Dr. Shilajit Borua for his technical support. Thank you, Dr. Prosenjit Das, our HOD Microbiology, for your leading role throughout time. Thank you. Thank you, everyone.
Thank you very much.
Sir, this session is now over. So over to Moboni.
Yeah.
Thank you, ma'am. With that, we come to the end of session 1 of this webinar series. I, on behalf of the organizing committee, apologize for all the technical issues faced today. I would again like to thank Dr. Prosenjit Das, Dr. Shudeep Shamadar, Dr. Gaidi Shahakesh, Dr. Shilajit Borua, Digongana De, and definitely Dr. Ranodhir Chakraborty for this wonderful session. Thank you all my participants for your patient hearing and interactions to make this session a success. Kindly fill in the feedback form already shared on YouTube to receive your certificates. We hope to connect with you all again for session 2 today at 7 p.m. I would like to thank PD sir for trusting me with this job. And with that, I declare the session closed. Thank you all.
Thank you, sir.
Thank you. Thank you.
Thank you.
Thank you.
ডিপার্টমেন্টের যারা আছেন বাদ দিয়ে অন্যরা কাইন্ডলি একটু লিভ করবেন। আমরা একটু ছোট্ট করে একটু মিটিং করে নেব। Moboni, you may leave, okay, and Digongana also.
Sorry sir, আপনি কি কিছু বলছিলেন?
You may leave now, okay?
Okay.
You conducted well.
Thank you, sir.